Hello and welcome to the second video of Interesting Science. My name is Alex, and today I'll be showing you how to make a slide of red blood cells and see them through a microscope. There's only a few things we need to do this. We're going to need a microscope that has a minimum magnification of 400 times. We're going to need 2 microscope slides and a lancet to collect the blood sample. Before we start, let's go over some basic information about red blood cells. So red blood cells are the way that your body gets oxygen to all the other cells in your body because they all need oxygen to function. Now they have a protein in them called hemoglobin that carries this oxygen. And this hemoglobin is what gives your red blood cells the red color and subsequently your blood its red color. They have a very distinctive biconcave dish shape that looks almost like a donut that didn't have a hole punched all the way through it. And they're constantly produced by your bone marrow because each cell only lives for about 120 days. So now that we've gone over that, let's get into it. So in order to get a clear view of the individual red blood cells, we're going to need to do what's called a blood smear. And the first step in getting a blood smear is getting the blood sample. So you're going to use your lancet and you're going to get 1 drop of blood, and you want to get that 1 drop of blood just on the edge of one of the slides, and it should look just like this, just 1 drop of blood on the edge of the slide. And then I'm going to zoom in to show you what you're going to do next. So here I have my slide with the drop of blood on it, and next thing is you're going to take your other slide and you want to take it, and at an angle you want to touch the other drop of blood so that it sticks to the second slide, and you're going to drag it and smear the blood in an even thing. So see how I touch the blood and it sticks to this one, and then when I drag this slide along it creates an even smear of the drop of blood, and that's going to spread the red blood cells out. So when we put it under the microscope, we'll be able to see them clearly. So now I have my blood smear on the microscope, and I have it pointing at one of the thinnest points on the smear. I have my camera up to the eyepiece of the microscope, and let's start at 40 times zoom and see what we can see. So if I get this to focus, there we go. So this is the blood smear at 40 times zoom, and you can kind of see the individual cells. If you look at those dots, each one of those is one individual red blood cell. But right now it still kind of looks like one big sea of things. So I'm just going to zoom it into 100 times. Okay, there's 100 times zoom. If we focus this, you can see the individual cells a little more clearly, but it's pretty zoomed out, and it just kind of looks like a big sea of them. So now I'm going to go into 400 times zoom. And let's see what we see here. This should be interesting. So we focus this now. There we go. So there we go. So this is the red blood cells at 400 times zoom. And you can very clearly see each individual cell and the biconcave dish shape. Looks like a little circle, and it has a little indent in the middle. So yeah, that's it. Red blood cells at 400 times zoom. Pretty cool. One quick tip for troubleshooting: if you are focusing your microscope at 400 times zoom and it's looking like this, all clumped together, and you can't really see the individual cells, you either got a bad smear, it wasn't thin enough, or you just need to zoom in or focus on one of the thinner parts on the smear. So if I take mine and move it farther away to one of the thinner parts and then focus it, you can see the individual cells much clearer. But that's it. Thank you for watching.